Hey guys, how's it going? Michaela, are you hooked up to audio?
Yeah, I got it.
You look like you're in a basement or something.
I'm under my bed.
Oh, okay. Okay, that's what that is. Got it. I'm like, okay, that's unusual. I am clearly still in my office. You guys are probably going to be thinking, oh my gosh, does he ever leave his office? Because that's the only place we ever see him. Hey, Dr. Judd. Dr. Judd, you look like an air traffic controller.
Yeah, I still haven't finished troubleshooting my new wireless headset, so I'm sticking with
Oh,
the
yeah.
one. Okay.
Well, how are you guys adjusting to the new reality?
Not yet.
All right.
So good.
What?
Somebody said what? I thought I heard a what. You're not Zoom fatigued yet, are you? Okay, a couple of housekeeping things here at the beginning. It looks like about 6 of you still haven't taken the pre-test. That's gonna expire at 5 o'clock this afternoon, mainly because I have another class that I need to give a pre-test in, and the platform only lets me run one test at a time. So I need to also pre-test my other course. So if you guys could see to that, one of you started the exam, but only did, I think, 7 questions. You can go back and restart the exam again, and we'll delete the one that's the incomplete one. So if you're in that category of didn't finish it and had to log off for some reason, or just haven't gotten around to it, please do that sometime today before the end of the day. A couple of you have made appointments with me for the Get to Know You sessions. Dr. Judd, have you met with anybody?
I've not. I was in my office all afternoon yesterday. I've been watching my email.
Okay.
So I'll watch for you all.
All of mine have been done on Zoom so far, so if you want to meet that way, that's totally doable. So just once again, that didn't have to be done this week. We've got, I think we said 2 weeks to get that done.
Yeah.
But yeah, just make sure that doesn't fall off of your radar. Dr. Judd, did you have anything else?
For housekeeping, just wanted to make sure everyone got the new link, and it looks like we've still got some people logging in.
Oh yeah.
So once we hit a good attendance there, I think we'll be good to go.
Okay. So if you look in the syllabus and remind yourself that participation is part of your grade, one of the things that we've done in the past is we've divided the class up into teams kind of along the houses of Harry Potter. And we keep you in those teams throughout the semester. And as you participate in class, we keep track of the teams. And so what I needed to do was get you into breakout sessions where I could control who's in which breakout session, which basically means that I had to delete the old meetings and set up a bunch of new meetings because you have to do it at meeting setup. So the way we do this in the past is we used to have big sticker boards, big Post-it notes on the wall with your team name on there. And as we go through the semester, we would make hash marks when your team is participating and things like this. And the nice thing about that is that everybody in the room knew where each team stood. And we knew everybody's name was on the Post-it notes, so we knew who was in each team, etc. And that's going to be a little more difficult to do. So I think what we're going to do this semester is every Monday, we will update on the Moodle site what the team standings are in terms of participation points. And so today, we're going to try the breakout rooms. So I just set them up. We're going to try and break you out into those so you can work in your groups, and then we'll get everybody back together periodically. So it'll be a set of going to a breakout room, doing some things, coming back together, sharing, going to a breakout room, doing some things, coming back and sharing with class. I'm not sure how smoothly it's going to go because it's the first time I've ever used this functionality. So bear with me if it is a little rocky here at the beginning, but this is a thing that we do with previous semesters a lot, which is break you up into small groups and have you work collaboratively on things. And so we want to retain as much of that as possible. As I understand, in your breakout rooms, you can share your screen with other people. So if you need to do that in order to get a point across, then use that functionality to do that. What we want you to be doing is talking to one another, sharing ideas, and then coming back with the products that we've asked you for when we put you into those breakout rooms. When we do participation, I've made myself a chart, actually. It has all of your names on it, and it has each day of the semester. And then I've got you color-coded in my computer, even though the colors don't appear on this sheet. They're color-coded, and they're color-coded by which group that I've put you in. So I have one of these that it'll be sitting on my desk the whole time. And as you participate, I'll be adding checks to this just as if it was a Post-it note in the classroom, so that we can keep track of this. And once again, I'll probably just take a photo of this every week as we go through the semester, so you can kind of see how you're building up participation points by team. You'll get to know your teams here in a few minutes when we break you out. So
sometimes while we get in the flow of things when we're talking, sometimes the other one will pay attention to who is, so we can fix that. There we go. One of the advantages of having two people on this is that if one of us is kind of in the flow and there's some participation, the other one can check, so we can make sure that we do get your participation and that it counts.
And Dr. Judd, I'm happy to share my thing with you if that would save you some time and effort.
And the other thing is, when we get into the breakout rooms, the two things to think about is if you are working on your iPad, you should have the opportunity to use a whiteboard. So in case you want to share your screen in that way, you can. If drawing helps, it helps me think. So that's just a reminder you have that opportunity. And we get into the breakout rooms, you should see a pop-out window that puts you into your breakout room. If you don't click that, you're just going to be all alone here with Dr. Kowinski and myself, and not in a breakout room. So watch for that. Make sure it's not hidden behind a bunch of other tabs or something.
And we have the ability to bounce in and out of breakout rooms. So in the classroom, what we basically usually do is we circulate among the groups. We bop in, listen for a little while, maybe ask a question or two, and then we bop out, go to another group, listen, ask questions. And so we're constantly moving around. Expect to see us pop in and pop out. If we don't say anything, we just come in, we've listened, and we left again. But expect us to also occasionally ask you questions designed to kind of keep you moving in the direction that we want you to be going. So that's once again a kind of thing that we've always done when we're together in a classroom, and we have the ability to do that now on Zoom as well. So did everybody look at the Coot photos? Okay, I'm seeing a lot of noddings of heads, good. So here at the beginning, what we're going to do is put you in your breakout rooms, and I have 3 tasks for you. Can you guys see the board on the back, behind me? No, you cannot. Who's that who cannot? Ryan, is it just too small?
I mean, I can see the board, I just can't read it.
The word.
Okay, give me a minute and I will move closer. It's always one of my concerns. So now can you read it? Okay, so in your breakout rooms, I want you to do 3 things. First thing I want you to do is to set up a time to meet over the weekend. This can either be in a socially distanced environment or in person, or you can do it through any sort of online communication that you want. So the first thing you should do is get to know your group members. So figure out who all is in your group. And I'll post these group memberships on the Moodle site later on today. So get to know who you are, just introduce yourselves briefly. set a time to meet and set up a group identity. So in the past, we've had people just choose a group name, and then we just kind of refer to you as a group by that group name throughout the semester. That shouldn't take you too long to set up a time. Then I want you to all share your COOT observations with one another. And as you share your observations with one another, be listening to other people's observations, because what I want you to come back with is, for each group, I want the 2 best observations that came out of your group work. So, and what I mean by best observations is, what are the observations that lead to other questions that you might have about the biology, the behavior, the ecology, the evolution of these birds? So you might make the observation, oh, these are water birds. Well, why do we know that they're water birds? Well, because they're sitting there in a pond of water. That's really evident from the picture. But just acknowledging that they're water birds, that doesn't lead me to think, well, why are they water birds? Well, there are a lot of water birds out there, and that's just, in some cases, like all the ducks and geese are water birds for the most part. There's not really a compelling path of inquiry that goes down that path. There are other aspects of them, though, that do send you down compelling paths of inquiry, and those are the kind of observations that we want to concentrate on. Everybody clear on what the job is? Okay, we are going to send you to your breakout rooms for about 15 minutes. Does that seem about right, Dr. Judd?
Yeah, I think so. We'll bounce around and keep
Okay.
track. But yeah, about 15 minutes is good.
Use the chat function to tell me if something's going wrong, but here we go, into hopefully breakout rooms. See you in 15 minutes or so. Breakout rooms not started. Dr. Judd, have you done this in the past?
I am, and I'm looking.
What?
The little button that I'm used to seeing is not here.
All right, hold on. Okay.
Checking.
The unfortunate thing about this is it's impossible to do this when you're on your own, because there is nobody else on there with you on Zoom.
Account. Here we go.
Unfortunately, you guys are experiencing our learning curve with the technology.
I've done breakout rooms many times, but not on a pre-set up meeting. I don't know why that would be any different. Account settings, breakout room option, make sure it's activated.
Yeah, I'm headed
already
that
there?
way myself. Settings.
Enable.
Wow, I did not know there was a settings tab. And well, I am allowed to do breakout rooms. It is enabled on my machine in theory. Okay. All right.
There's always been a breakout room button down here at the bottom.
Well, I have one on mine, but when I go to it, it says there are 6 breakout rooms. Oh, it didn't do the pre-setup. That's weird. Okay. All right. Give me a minute. I'm going to have to set it up on the fly, which is what I was trying to avoid earlier. Audrey, Brandon. Traveris. It's not here. Cody. Joseph. This is also the other reason it's important to be in class, because if you're not in class, your group ends up being on the small side. Riley, Alex, Grant, Keaton, and Trang, Paula.
Oh, would you like me to talk to them a little bit about what success is going to look like?
Yes, you can do whatever you want while
Yeah.
I'm dealing with this, because this was not what I hoped would happen.
So as we
Go
get
ahead.
started, with this activity and the next few that we're going to be doing, we want you to think about what success looks like in this class, because it might be different than what you've experienced in previous science classes. As we talked about last time, we're not going to be focusing. One of the goals here is to teach you how to think like a scientist, not just learn a bunch of biology and ecology facts. So in that case, success is about learning how to think and learning new things in a way that might require you to unlearn things that you thought you knew. It means recognizing assumptions that you might make when you're making observations, and then proceeding from the observation stage to the rest of the scientific method, developing a question, developing a hypothesis, examining data, that sort of thing. So by the end of this, what we want you to be able to do is to describe the scientific process. So be able to talk in maybe a more sophisticated way when you're asked to describe the scientific method than if you were to just Google it and find the list of steps. We want you to be able to think through that and how it's maybe applied differently in different contexts, or how it's operationalized or executed in different contexts, I should say. We're going to distinguish between trivial and non-trivial observations and questions, which Paul was just telling you about when he was telling you to get a sense of trying to rank your observations as you compare with your group. And we'll talk about what we mean by trivial and non-trivial questions and where you can take them going forward.
Have the room set up whenever you're done.
Okay. You're going to consider the importance of careful and thoughtful observations, which you'll recognize from the stuff you hear about critical thinking in other classes. So observation is going to be an important part of that, and that's why we're starting with this COOT, COOT activity. And then you're going to learn a little bit more specific stuff, like the difference between control and treatment group, which we'll get into in a little bit, and the difference between different kinds of experiments, some new vocabulary like mensurative and manipulative experiments. So these are the things you're going to learn in the next few classes. So there's your preview, and now we'll get you into your small groups and talking about your observations.
All right, here we go.
Oh, good.
Oh, they all disappeared. Yay.
Excellent.
Devin, you did not disappear. There he goes. Yeah, so all my efforts at setting them up in advance were for naught.
Well, it worked out. Yeah, let's see. Now I got to make sure that I was just looking at this too, and I got to make sure that I can access these breakout rooms.
I was hoping you would be able to because you're the co-host.
I fully expected to be able to. I thought I could. I've had multiple people join breakout rooms before at a meeting we had back in May, and now I'm not seeing it. So let me see if I can figure that out.
Okay. It's weird because you appear as a co-host, don't you? Or do you?
It says co-host me. Yep, and host you. Yeah, I see
Okay.
it.
All right. All right. Well, I'm gonna go pop in a room.
Okay.
I'm gonna just start at room 1 and work my way down.
Okay, sounds good.
All right.
What time on Sunday do you guys want to?
I'm cool anytime on Sunday.
I'm cool.
Same.
Do we want to do like in person type thing or just online?
I think in person will be cool.
Okay.
If you guys are okay with it.
I'm cool. Let's do like something later so it's not like super hot. So
Okay.
maybe around like 6, 7-ish around there.
Or early. I usually do yard work in the evening, and it's hot at 6 still.
Really?
Yeah.
Damn. Okay. So we can do early then.
Nathan.
So, Paul, it seems you have to assign me to a breakout room, and then I become free to move around, but not until I'm
Okay.
assigned once.
I will assign you to a breakout room.
Mm-hmm.
Nathan Judd I'll assign you to room 2, and I'll go to room 3.
Sounds good.
Similar to your both of your observations.
Me too.
I think I like the one that Cameron said about the eyes, how the younger ones have different color eyes than the adult cahoots. We can use that as one of ours.
Sure.
And Casey, I like your observation about the mother coot having the grass or leaves in her mouth because that showed it might be herbivores.
Okay.
And then when do you guys want to meet?
I can meet anytime on Sunday or early in the day on Saturday for the weekend.
Probably Sunday works best for me too. Anytime, I think.
Okay, we can do Sunday.
All right, where do we want to meet on Sunday?
It's the DLC.
Sure.
Okay.
See what time?
Whatever works best for anybody, because I don't have anything.
Would you rather do? Okay, which ones do you guys want to put in the chat?
So you got the message.
Probably the tip of the.
Yes.
Okay.
Probably the tip of the beak, the different color in that, because that's kind of hard to see.
Do you guys know anything about birds? What is the tip of that beak typically doing?
Cracking the shell of the egg.
Yeah, it's an egg tooth. So it's good that you observe that. Why is that maybe a less useful observation than maybe some other observations? Hunter can answer, or somebody else can.
I mean, I guess like with that one, it's like a very simple thing that like we know the use for. It's not something that like changes as much as other things.
Yeah, so
like
we can observe it, but when we see it, we know what it is. It's the egg tooth. It's a hardened portion of the bill. It's going to fall off in a week or so, and we already know what its function is. We know why it's there. So once again, that's an observation, but it doesn't send us down a path of inquiry that leads us to something that we don't already know. And this is what we do. We lurk, we come in, we ask a question, and we leave you.
It's a good idea.
Yeah. So what dorm are you guys in? Like we can meet in the lobby or something.
I'm in Eaton.
I'm also in Eaton.
Okay, I'm in Mathis, so I can just come over to Eaton.
Okay. Yeah, we have like a main lobby on the second floor or the first floor.
Okay, cool.
What time do we want to do?
This class gets out at what, 2:15?
I thought it was
Oh, no,
1:35.
1:35. That's right.
Yeah.
I don't know why I was thinking 2:15.
Yeah. So we can just meet like right after this one.
Yeah, that works.
Okay, sounds good. And then are we missing anything else?
We need to put our 2 best observations in the chat.
Yeah. Do you guys
Did you
have
already
any?
discuss your observations?
Yes.
Yes, we did.
Okay.
Yeah, I did not even think about the beaks. I was thinking, yeah, I did not even notice that. So I thought
What's the
that was pretty
observation
good.
about the beaks?
That they had black tips on the ends, or the babies did, and then the one with the mother had a white tip.
Mm-hmm.
Ah, so that white tip, do you have any idea what it might be?
Could it be like residue from something it ate?
Nope.
Does that have to do with
Yes.
gender?
No, it doesn't do that either.
Is it maybe
The
to
one-
like draw the attention of fish or something?
Nope. So that one that's with the mother is evidently a younger chick than the one that's in a group of 4. Does that give you any hint?
Maybe
The reason I call attention to this, what? Go ahead, Daniel.
I was gonna say like maybe it's malnourished, and so it's like losing pigment.
No, they hatch with this.
Okay.
It's there when they hatch.
Oh.
Okay.
So it's there from hatching, and then it drops off.
Okay.
Do you know what
So
what
is
it's
it
used
like
for?
the leftovers? Oh, what it's used for? For breaking the egg, I'm assuming.
Yeah, it's called the egg tooth. So all
Okay.
birds have it. It's a little white thing. It's hardened because the rest of their body is actually still pretty soft when they come out of the egg, and they start to harden up after they come out. And so it's a little hardened tip of the beak that allows them to break through the egg. So it's a good observation. It's a really detailed observation, but it's not particularly a good observation from the standpoint of asking questions about, because we know what its function is already. And so we already know quite a lot about it. And we're looking for observations that lead us down paths of inquiry where we don't know what the answers are. And I realize the 3 of you guys didn't know what the answer to it was, but with an hour of research, you would have figured out what it is. And then you'd be like, okay, well, that's already known. And then you've been at a dead end. So, all right.
I did have a question about why, or I guess like the observation that, what's her name, Michaela?
Michaela.
Is that your name? Michaela, okay.
Yeah.
How the adults like form the pigment, the red pigment in the eyes.
Right.
I thought.
So there's variation in eye color.
Yeah.
That's something that we probably don't know much about.
Mm-hmm.
I'm going to head
Okay.
to another room. Oh, you guys are all silent.
We're writing.
Okay.
We're trying to formulate our ideas.
Okay, good. Pausing to think is not a bad thing.
I'm on my phone, so it's not really easy to type.
All right, what do we want our second, like?
Eat for food.
Speak up.
So
Speak up.
So baby coots, they compete for food, and it's really competitive, and the younger ones will die off. And so the brighter ones and more vibrant looking ones, their parents will favor them more, and so they will get fed more often.
Okay. All right. What other ideas did you have?
Well, about the bright colors, I thought
Mhm.
it might be so like they're easier to find by their mother. Like if they swim off, it's easier for them to be seen and recognized.
Other ideas?
I thought maybe it might be something with their ecosystem where like they live in an area with bright flowers that they can camouflage well with.
Okay. Other ideas?
This one is kind of really out there, but kind of like how flamingos get that pink pigment, maybe it's based off of something that only the babies are fed.
So it might not have any function at all. It's just an artifact of their diet. That's a mistake that sometimes biologists make. Biologists sometimes make the mistake of thinking that every aspect of an organism's biology, every aspect of their morphology has to have some function, but sometimes things are just artifacts of other things. And that's also a perfectly reasonable explanation. So we got 5 ideas that might explain this. Are there any problems with this list as a whole, as you look at one explanation versus another?
Well, if I can jump in, I feel like camouflage and defense like coloration are kind of mutually exclusive.
Okay. So camouflage is the act of being hidden, but defensive coloration is the act of making yourself known to the world. And so certainly those are 2 hypotheses that are mutually exclusive. So it couldn't be this and this. Is there another one that is also mutually exclusive?
Possibly 2 and 5. Babies compete for food, but maybe I was reading wrong.
Well, yeah, I don't see those as being as
never
much
mind.
of a, those could both perfectly exist well together.
I think that 3 and 4 are too, because if it's easier for the parents to find them, then they wouldn't be able to camouflage.
Yeah, if it's easier for your parents to find you, it's also easier for a predator to find you. And so we can look at a list of potential hypotheses and see that hypotheses have conflicts such that if one of them were true, it would automatically negate the others. Does anybody know about the distribution of aposematic coloration out there in the natural world? What kinds of organisms do you think of when you think of aposematically colored organisms?
Have any of you encountered aposematically colored organisms that are obviously warning you to stay back?
Oh yeah, I saw the snake that was like orange, white, and black, and it was pretty cool looking, but I didn't get close enough to like really examine it.
So coral snakes have aposematic coloration. Somebody mentioned poison dart frogs have aposematic coloration. So amphibians, butterflies, the monarch has aposematic coloration. It has cardiac glycosides in its body that it gets from milkweed plants.
I was going to say the blue-ringed octopus.
Blue-ringed octopus, certainly. Somebody else said something?
Yeah, I said water moccasins.
They have banding, but sometimes water moccasins are just slate black. What is their aposematic coloration? What do they do?
Yeah, like I had seen one that was all black too. It had yellow diamonds on it. It was like all black and it had yellow diamonds. It was weird.
Oh, really? Oh, interesting. Water moccasins, cottonmouths, they're called cottonmouths because when they're defensive, they open their mouth and their mouth is bright white, whereas the rest of their body is relatively dark. So it's kind of a behavioral and a color aposematism. What other insects can you think of that are aposematically colored?
Spiders.
Maybe some bees.
I'm thinking more of bees. I'm trying to think, most of the really brightly colored spiders that I know of, what's the other function that bright coloration sometimes serves? And this is true in birds as well. Attracting mates. So most of the brightly colored spiders I know of that I can think of, it's more about attracting a mate than warning predators. It's not to say that it couldn't actually do both, but in both those cases, it's a coloration that is designed to attract attention, in one case to attract the attention of a mate, in another case to attract the attention of a predator to say, hey, don't mess with me.
What about mushroom? Like some of them are not like toxic at all, but it looks really bright, so we think that it's toxic.
Dr. Judd, do you know much about the colors of poisonous mushrooms? This is way outside of my field now.
Yeah, it's all over the map. So it's an intuitive hypothesis that a lot of people have, but when you look across the diversity of mushrooms, we see both really vibrant toxic ones and really dull toxic ones. I don't know if anyone's really looked at that, but that's interesting. Interesting reminder of something to look into.
So what's a group of organisms that we haven't mentioned yet?
Mammals.
Mammals? What other group that was relevant to the current discussion?
Birds.
Birds. Birds are kind of absent from this, aren't they? A lot of amphibians and reptiles, a good number of insects, marine organisms. There are all sorts of examples of aposematism, but we struggle to find examples of aposematism within birds. And there are only about, Dr. Judd looked this up, and there are only about 5 or 6 birds that are known to be toxic. And when we were looking at this last week or earlier this week, I guess it was earlier this week, at least a couple of them that we saw pictures of, they appear to be aposematically colored, but it's a relatively rare thing to have happen in all of the almost 10,000 species of birds that are out there.
It only happens in the most species-rich places in the world where there's so many birds. Maybe the odds of evolution discovering aposematism in one species here or there may be achieved, but across all of North America, there are none.
Yeah, all of the species occur in the tropics. So of all the of the approximately 10,000 species of birds, we only know of it in a handful of bird species. So it could be that that's the case, but once again, looking at it phylogenetically, it would kind of argue against that. As it turns out, that's not the question that these researchers addressed. It happens to be this idea that babies are favored by the parents for provisioning if they are brighter colored. So who was it? Which group had this hypothesis?
That was me.
Okay. All right. Who's that? Grant. Okay, Grant.
Grant. Yeah.
So what we want you to do now, and I'm going to have to see if I can get you back into breakout rooms without going through an onerous task. Oh, you're all still in your rooms. So what I want you to do, really probably for the remainder of the class, because it's going to actually probably take you longer than 10 minutes to do this. The researchers that this case study is based on had this hypothesis of parental favoritism. And basically the idea is that brighter birds, brighter offspring, get more attention. And this could be all kinds of attention. A lot of times the attention that you guys have mentioned is food, but it can also be protection from predators or incubating the offspring after they're hatched. So what I want you to do, what I want you to begin doing in this next 10 minutes that we have left in class is get in your groups and start thinking, what kind of experiment would we do that would allow us to test whether this hypothesis that brighter birds get more attention would be true. And what I want you to think about is, are you going to do something that would manipulate the birds or the environment or the parents or something like that? Or are you just going to make observations of the birds, and how would those observations be? How would you determine who's brighter or who's not brighter? I want you to think about the kinds of variables that would tell you whether or not one chick was receiving more parental care than another chick. So what would your experimental design be? What kind of experiment would you design that would get at whether or not this particular explanation actually is the right explanation. Dr. Judd, do you have anything to add to that before they go?
Think about what kinds of data you would collect, right? Whether that's after manipulating or a kind of observation, and what do you have the capacity to measure? If buy all the supplies you need to do that measuring.
And don't limit yourself at this point to a budget or anything like that. Assume that the National Science Foundation thinks you're freaking brilliant and has given you all the money in the world to do this. All right, we're going to be bopping in and visiting you, so we'll see you in your rooms. Devin, why are you unassigned?
It's because the first time I had unconnected from my hotspot, like it booted me off, and I had to join the whole classroom, so I wasn't able to get back into my other one.
Okay, I'm going to put you in your room where you'll normally be.
Okay. I see it. Thank you.
And we'll have to troubleshoot a little bit, because I think I need to be an alternative host or something, because now I can access breakout room 2 and come back here anytime, but I can't get into the others. I don't know if you can assign me to multiple breakout rooms. I was trying to find that out last.
Yeah, I didn't.
I saw room 2, so I jumped in on 2. I guess I was right.
No.
I have no idea how we would do an experiment.
You could almost like get like an adult or something, and I feel like you could have like fake babies or something, and you have like maybe different stuffed animals and see which one it goes to more often.
Like that.
Mm-hmm. And then you could almost do that, like you could have the same colored babies and everything, like the same stuffed animals, and
wouldn't be actually hurt or anything.
Yeah.
No, yeah, that'd be
That's
good.
good.
And then we can see if the ones with the brighter color like develop faster, or if they just way more get bigger than the colored one.
I was thinking the observation route would be pretty useful too, because then we could look at like an entire population of coots, and we'd have a lot more data than just like working with one mom and like one group of babies. And then I'm sure that there would be like weird instances where we get like a really bright coot that like the mother or something like only gives attention to. I think that would be kind of cool. So I think if we're doing an experiment on this, we'd have to find like their mating season, so we could know when they're gonna lay eggs, and then just watch as many as we can. But I don't know, like we'd have to develop like what we're observing, like what makes a coot, like, I don't know, more attractive to the mother. Is it just being brighter, or is it like other factors involved? So there's lots of questions.
Keep in mind what the hypothesis is. The hypothesis speaks strictly to coloration, so that's kind of a limitation of kind of how we frame the question.
Okay.
And also to speak to another thing somebody else said, the idea of doing more than one family is good. It's what we call in science replication, so that you have lots of different instances of the thing that you're looking at. That's always a good thing to have.
So when you say coloration, does that, if it's just like a more vibrant red, does it, are you talking about like different colors or just different?
Yeah,
different
so
shades of red?
one of the things that you would have to do is decide whether you're going to stick with the normal range of coloration that is there, or if you're going to do something that drastically alters that. But certainly you wouldn't want to make them blue, because blue isn't a color that any of them are, so you would want to stick with their natural coloration.
So if we wanted to, let's say, have like 2 or 3 of them be like the red color, and then we pigment one of them to be like black, like their mother, would that be like not okay?
No, you could totally do that. You would have to find a way of doing that that was safe for the chicks and all that. But yeah, that's a manipulative experiment where you're manipulating the variable of interest, which in this case is coloration. All right, I'm going to go join another room.
Yeah. We said that we would have to like measure the amount of time spent with the parents and versus like each sibling and kid, and mostly just make observations about which one is brighter and see if they favor that one or not.
Is this directed at me?
Yeah.
Okay, so what are some questions that you need to be asking yourself about that approach?
See what we determine as brighter, like how, like a set of standards we can apply to which ones we consider.
Yeah, how do you determine brightness of the coloration of a coot? That could be very tricky and difficult to work out. One of the reasons I don't like working with color myself is because it's something that's sometimes very difficult to quantify. But there are ways of quantifying it. There are plenty of people who do work with color. You would just have to do some research into figuring that out. So you're proposing what we would call a mensurative experiment, where you use the natural variation that is out there to set up your experimental groups in a sense, and then use that variation to look for differences among the groups. So you have brighter chicks that are naturally brighter than other chicks that are naturally not so bright, and you would look for differences among those groups. If you go back and look at the chicks, you might see what some of the complications of doing that would be, especially the group of 4. I'm going to go join another group. Everybody here? Not everybody's here. Okay, the group is repopulating now. All right, can everybody hear me? Or any of you for some reason. So we're out of time, basically. That's why I called you back. You to, well, what is up? Yeah, for some reason. I'm not sure why it's not interfacing. It's never done this before. What we want you to do as homework for Monday is, as you're meeting with your groups to get to know one another, you now have another task. It sounds like not all of you have fully worked out what a good experiment would look like. So what I want you to do also when you're in your groups, what the experiment will look like that you would propose doing. And then what I want you to do is elect one member of your group at the beginning of class on Monday to present in a very brief form, probably about 3 minutes, what you plan on doing, what you would do if you were going to do this experiment. You would measure what observations would you be making. Are you going to be manipulating the birds? Are you going to be doing what we call a mensurative experiment? All of those things. Organize that into like a little 3-minute spiel. We'll hear from each group, and then what we'll do is we'll tell you about what these people done, and then we'll start thinking about what they did, what they found, and what they kind of concluded, and that's what we'll be doing on Monday. Is everybody clear with that? Hold on, Dr. Judd sounds like he's trying to. Dr. Judd?
Outcomes are. What might you see, or what might you not see, and what would those things mean?
I have a question.
Yeah.
Will we be able to do like additional research outside of the 2 pictures that we are given, or?
No.
In this case, don't do that.
Okay, sounds good.
Yeah. Because you're going to stumble upon the paper, and we don't want you to come to the activities on Monday with any knowledge of what the answer is.
Good question, but yeah. And normally we would encourage you to go and do research, but just not this time. All right, everybody clear? Okay, well, email us if you have any questions, and we'll see you guys on Monday.
Thank you.
Thank you.
Hey, Nathan, you have class after this?
I do in about 10 minutes. Yeah.
Okay, all right, I'll talk to you later.
Okay, you might notice that if you do the little up arrow on the microphone button, you can do the headset, the speaker and the microphone are controlled separately. I was having trouble with that with my new headset.
Yeah, it's weird because I've never had this happen. Like all through the spring and summer, I've never had this happen. So
did you ever use breakout rooms?
I didn't ever use breakout rooms.
I think that was part of my trouble the other day.
Okay, all right. Well,
that's what we'll have to research, I guess.
Clearly we have some learning of Zoom to do.
Yeah.
All right. Good luck today.
Yep, thanks.
Bye.